Today we are going to discuss a very important topic in biochemistry known as carbohydrate metabolism. As you know, every living cell requires energy in order to perform its functions such as growth, movement, synthesis of molecules, and maintenance of cellular structure. But where does this energy come from? The energy that drives all cellular activities is obtained from the chemical bonds present in the food we consume. Major nutrients such as carbohydrates, fats, and proteins are broken down through various biochemical pathways, and the energy released is stored in the form of adenosine triphosphate, or ATP. ATP acts as the universal energy currency of the cell. The process through which carbohydrates are broken down and converted into usable energy is called carbohydrate metabolism. Let us begin by understanding how food molecules are processed. The food we consume contains proteins, polysaccharides, which are carbohydrates, and fats. Carbohydrates are first digested into monosaccharides like glucose, absorbed into the bloodstream, and transported into cells for energy processing. Inside cells, glucose undergoes glycolysis to produce pyruvate, two ATP molecules, and NDH as reducing equivalents. Pyruvate enters mitochondria, where it is oxidized to acetyl-CoA, releasing CO2 and NADH. Acetyl-CoA feeds into the tricarboxylic acid cycle, generating more NADH and FFADH2. Electrons from NADH and FADH2 drive the electron transport chain, creating ATP. Digestion breaks polysaccharides into glucose. Enzymes like amylase and brush border enzymes finish the job for absorption into the bloodstream. Glucose is transported into cells via GLUT transporters, then phosphorylated to glucose 6-phosphate by hexokinase, or glucokinase using 1 ATP. Glycolysis splits glucose into 2 3-carbon molecules through 10 enzyme steps in the cytosol, yielding a net 2 ATP and 2 NADH per glucose molecule. Key regulatory steps occur at hexokinase, phosphofructokinase 1, and pyruvate kinase, responding to ATP, AMP, and fructose 2,6-bisphosphate signals. Under anaerobic conditions, pyruvate is reduced to lactate to regenerate NAD+, allowing glycolysis to continue in muscle and certain microbes, though ATP yield is limited. When oxygen is sufficient, pyruvate enters mitochondria to be converted to acetyl-CoA by pyruvate dehydrogenase, producing NADH and CO2. The pyruvate dehydrogenase complex links glycolysis to the TCA cycle by forming acetyl-CoA, and it is tightly regulated by energy status and covalent modification. Acetyl-CoA combines with oxaloacetate to form citrate, beginning the citric acid or TCA cycle inside the mitochondrial matrix. Each TCA turn releases 2 CO2, generates 3 NADH, 1 FADH2, and 1 GTP, which can be converted to ATP by substrate-level phosphorylation. The TCA also provides intermediates for biosynthesis, such as amino acid and heme production, linking energy metabolism to building blocks. Anaplerotic reactions refill TCA intermediates. For example, pyruvate carboxylase converts pyruvate into oxaloacetate to maintain cycle function. Regulation involves substrate availability, NADH levels, ATP, ADP, and calcium signals within mitochondria to match energy supply and demand. NADH and FADH2 donate electrons to the electron transport chain embedded in the inner mitochondrial membrane. Complexes I through IV transfer electrons while pumping protons into the intermembrane space, creating an electrochemical gradient. This proton motive force drives ATP synthase, which synthesizes ATP from ADP and inorganic phosphate as protons flow back into the matrix. Oxygen serves as the final electron acceptor, combining with protons and electrons to form water at complex 5. Theoretically, 1 glucose yields about 30 to 32 ATP through complete oxidation, depending on shuttle systems and cell type. Uncoupling proteins can dissipate the gradient to produce heat rather than ATP, a process important in thermogenesis and metabolic regulation. Cells balance energy demand by switching between glycolysis and oxidative phosphorylation depending on oxygen and substrate availability. Hormones like insulin and glucagon regulate carbohydrate metabolism by controlling glucose uptake and key enzyme activities in liver and muscle. Genetic or acquired defects in these pathways can lead to metabolic diseases and energy deficiencies that affect many tissues. Examples include pyruvate dehydrogenase deficiency and mitochondrial disorders that impair ATP production and cellular function. Laboratory diagrams show carbon flow, ATP yield, and redox carriers. Animation helps visualize molecules breaking down into ATP for better comprehension. In summary, carbohydrate metabolism converts food into ATP through coordinated pathways: glycolysis, the TCA cycle, and oxidative phosphorylation. And that wraps up today's topic. If this explanation helped you understand the concept more clearly, make sure to like the video and subscribe to this channel for more simplified lab technology and radiology lessons powered by AI. We break down complex subjects in hematology, biochemistry, microbiology, pathology, X-ray, MRI, and diagnostic imaging step by step, so you don't just memorize, you truly understand. If you have any questions or want a specific topic covered, drop it in the comments below. Until the next video, keep learning smart, keep thinking critically, and keep mastering diagnostics.